No voy a darte la charla en español porque yo creo que sería más entendible para mí si de vez en cuando hablo inglés y tal vez mantenerlo así. So thank you, everyone. I will do the rest of it in English. So if you were going into panic mode, no need for that. So the work I'm going to talk about today, first of all, can you see the cursor? Yes? I can see where I'm pointing. I will assume that you can.
You're good.
So this is work from Jack Samuelian. It began in my lab with Manami Rajadri Shaha when I was at Indiana University. It's turned into a multi-institutional collaboration. Dana Baum has done some just amazing things at St. Louis University. Xiao Hong and I are at University of Missouri, and Jack is a student in Dana's lab. And so that's, most of this work will be from his work. How do I advance this line? There we go, it's not working. There we go. Okay, había una vez. So we're gonna start off with a just so story, once upon a time. So I think we can all agree that RNA has been around for a very long time. If you were to trace back the origin of the ribosome, it would certainly go back billions of years, and that in most people's book is a long time. So it's clear that once upon a time there was RNA. What's less clear is whether once upon a time there was an RNA world. I'm not sure who took this photo of the RNA world organism, but there you go. And the idea is that somehow there was an RNA world organism, and through the magic of evolution, it became the life types that we're familiar with now. Now, we are empiricists in my lab. We do love theory, but we also want to put the pieces together experimentally. So, with that in mind, we are looking to build RNA enzymes for early life. And I have to, at this point, announce to the world what NASA announced just this week, that our team was selected for a NASA Interdisciplinary Consortium for Astrobiology Research. This is a multidisciplinary group of 8 institutions and 12 PIs, essentially to do exactly this, to build RNA enzymes for early life. And there is a postdoc position associated with this in my lab, as well as some others. So how does one build better ribozymes? Some of the features that we really need to work on include things such as multiple turnover, getting metabolic flux, small molecule and substrate specificity, much less trivial than you might think. Biologically relevant reactions and substrates rather than the toy reactions that we've been grown accustomed to. Push some of these reactions energetically uphill through coupled reactions, for example, sequential and pathways, and getting bioactivity out of it. So we're going to focus today on looking at uphill reactions, and specifically asking the question of, can RNA aptamers activate electrons to drive unfavorable redox reactions? So how do we think about this? It would be quite familiar to most people to think about coming down a slide. That can be your reaction, but you have to first activate the kid to get up there to the higher energy. And that is the analog of what we want to do. You can think of this as your electron running around, or rather, this is the willingness of a given flavin molecule to give up its electron. Somehow we need to activate that flavin so that it's much more willing to give up its electron. That's the goal. Or if you prefer to think of it, native peoples all over the world have developed simple tools to impart more energy into their spears. This human, for example, is using what's called an atlatl. And there's your spear, and they can chuck that spear much farther and with greater force. So we're going to build simple tools to chuck the electrons with greater force and throw them farther than they would go otherwise. Okay, so that's the goal. Use RNA. Can we use RNA to build a simple tool that will activate electrons so that they can do reactions they would not do otherwise. So here's some of the theory that goes into it. And the title says everything, really. Differential binding energy activates flavins. Flavins are great for carrying electrons around, but they are much more willing to get rid of their electrons in particular environments. And binding energy is the key. So let's start with flavin adenine dinucleotide in the top left corner of this thermodynamic cycle. It could either bind to an enzyme by coming down the cycle and then be reduced by going to the right, or it could first be reduced in its free form and then the enzyme could bind to the reduced flavin. Either way, the energy going around the cycle should be the same. If you lay out the delta G terms for going around the cycle in the two different directions and rearrange them, what you get on the left here in the blue box is that the difference in the binding energy should be exactly equal to the difference in the free energy of reduction for the free and the bound form of the flavin. So this is a principle that proteins are very well known to do. Many different proteins fine-tune the potency of the flavin that's bound to them, but no one's ever demonstrated this for RNA. So that's the background. Let's do some experiments. We used a fairly standard approach to in vitro selection for making aptamers. I won't go through the details right now. I'll be happy to in the Q&A if you wish. In brief, we took a 42N library, ran it over an ADP pre-column to reduce the fraction of the library that bound adenosine. Let it go straight from there on an FAD column, threw away what did not bind, and did a slow elution with FAD to capture what would elute with FAD. RT-PCR back into RNA with transcription, and then round 2. And round 3, 4, 5 did a bunch of rounds. 12 cycles later, we then exited the cycle for cloning. As we screened those aptamers, many of them preferentially bind oxidized flavins. And here's one example, aptamer number 12.29, as measured by inline probing, has a KD for FAD of 30 micromolar and for FADH2 of 500 micromolar. So somehow it's differentiating between the oxidized and reduced forms of the flavins. Now, these numbers were measured by inline probing. When we shifted over to the much more accurate isothermal titration calorimetry, or ITC, this variant of aptamer 12.29, this variant is known as aptamer X2B2, and this one has an even longer name. And we were able to get the dissociation constant down to the neighborhood of 200 nanomolar. This is for binding to FAD. Okay, so those are all very labor-intensive experiments. Jack came up with a really nice spectrophotometric way of monitoring binding, and it works like this. The free flavin has a spectrum that looks like what's shown in orange here, which is sort of the color of the flavin anyway. Once it's bound to the aptamer, the maximal absorbance shifts from 448 nanometers over to about 456 nanometers. At the same time, a new shoulder develops at about 482. So simply by looking at this spectral shift and the appearance of the shoulder, we can ask the question, what has happened to binding as we perturb the conditions? So we went through a bunch of different aptamers. Here's another one called F-test. It seems to bind okay. Here's another one called 27FAD1, which is shown in red, which really doesn't seem to do anything different from a negative control or the free flavin itself. So there you go. X2B2 had the greatest peak shift, denaturing conditions, and other sorts of things, ran it straight back to FAD, and we have now a quick method for monitoring binding. I'm going to run through the next several slides quickly. This is not really about how the binding works. It's just to show you we do some good science from time to time. So we looked at the magnesium dependence and showed that as we go from 0 to 1 to 2 to 5 to 10 to 30 millimolar, that the bound portion went up. As we shifted to different divalent metal ions, we found that pretty much all of them supported the binding, as long as there were some divalent metal ions there. This same aptamer, X2B2, tolerates pH 10 just as readily as it tolerates pH 4. That was quite the surprise. We can do melt curves by changing temperature. We can look at inner sphere and outer sphere interactions by substituting cobalt hexamine, which, by the way, does not support binding. So cobalt hexamine data are shown in orange here, no evidence of binding, strong evidence of binding with magnesium. Oh, and we looked at a bunch of different analogs of FAD. FAD is this full-sized molecule here, and it shows the the shoulder forms, the spectral shoulder forms. So that is bound by the aptamer. FMN, that's flavin mononucleotide, is also bound. Riboflavin is missing that terminal phosphate, and it binds just nicely. Even formal methylflavin, which is just the flavin ring and an aldehyde attached to it, even that shows the spectral shift. So the aptamer is clearly interacting primarily with the flavin ring itself. Okay, fun facts, just to summarize up to what we have so far. In fact, I'm going to skip the fun facts because I really want to get to the second half of this and look at these shifting of the reduction potential. We're going to use isospectic points for the next part of the assay, and I want to define what that means. If you have two compounds, A and B, that are interconverting in solution, but they both contribute to your absorbance, you can use isospectic points to monitor the ratio of those two. What you can do is to, an isospectic point is simply the point at which the absorbance of each of the two compounds is the same. The two forms of the compound is the same. So pure species A shown in red, pure species B shown in magenta. It doesn't really matter what the ratio is. The total concentration drives the absorbance at that wavelength. And that's what we're going to use soon. Okay, but before we get there, here's the redox assay itself. We used a coupled enzyme assay. Xanthine oxidase will take xanthine, shown on the left here, and molecular oxygen. There's the xanthine oxidase enzyme, and it will convert it to uric acid. If there's no oxygen around, then you can substitute a dye such as methylviologen, and it will use the dye as the electron sink. Still make the uric acid, but now it generates the methylviologen, which gives a bright color in spectrophotometry. There's some math you can do on that, but what is most important is that the y-intercept of the plot that comes out of it is the difference in the reduction potential between the flavin and your reference dye. So we're going to be looking at the y-intercept of some plots here in just a moment. And as that y-intercept changes, it means that the reduction potential of the flavin has changed. There's the dye that we used. It's called AQS. We can come back to that later if you want. So here are the data. Well, it's one batch of data. We have many batches of data. The isospectic point for FAD is at 335 nanometers. We use that to monitor the concentration of the AQS. And the AQS isospectic point is at 355. We use that to monitor the concentration of the FAD. Putting those two together, we came up with the equations that were there before, plotted those on these data, look at the y-intercept, and what you can see right away is that the free FAD and the negative control, that's the orange and the gray, they map right on top of each other, and the negative control RNA does not shift the reduction potential. This other aptamer called F-test1 barely shifts it, if at all. But these two, especially X2B2, creates a whomping shift that we can very readily measure what the shift in the reduction potential is. Plotting that onto, plotting the reduction potential, E0, for each of our different aptamers that we measured. We get no change for the negative, barely any for the F-test1, minus 12 millivolts for the X2B2, but the high affinity point mutation minus 43 millivolts. Now, to put that into context, here are the dotted lines show the shift of the Flavin reduction potential for two different protein enzymes. HPA reductase shifts the Flavin by minus 25 millivolts, and APPA bluff shifts it by minus 51 millivolts. So our aptamer is activating the Flavin to a degree that's right in the range you'd expect for protein enzymes. And with some more math, that corresponds to about minus 2 kilocalories per mole. Okay, so conclusions. We've built some simple RNA tools to impart more energy into the electrons that are bound by flavins, approximately minus 43 millivolts. And next up will be to build better ribozymes by rational engineering of the parts, drive reactions uphill, multi-turnover reactions, coupled reactions into pathways, putting them into organisms for model riboorganisms. And I'd be happy to connect with you through social media or other means. And therein, I think, is the end.
All right. Thanks, Don. We're a couple minutes over here at this point. So if anybody's got questions for Don, shoot them over to him in the chat, or as he has suggested, certainly reach out to him on social media, where he is very good about getting back, which I very much appreciate. Thank you so much. Interesting stuff, Don.